Buongiorno a tutti. Allora,
Good afternoon. Let me share with you, first of all, the history of this, because Paolo Antoniali addressed us and the Yeast Genetic Laboratory in Padua, at Padua University. So he addressed me and Dr. Sartori in order to perform a molecular analysis whose objective, in general, was that of sort of making order in yeast strains which IB manages directly, and also with a view of making order in the production chain of industrial yeasts. That was the beginning of our cooperation. We decided in a very first phase to go for a systematic genotyping activity, whereby we would identify strains of interest for IB and then use that information to identify the molecular identity of the raw material, the yeast, and also in order to have a quality control of the end product. This is the direction we went. Initially, we had to choose a way to identify the strains. To do so, we actually referred to the organization which is the reference for this, which is OIV. Specifically, we decided to select the amplification of delta sequences in order to discriminate and identify strains. We talk about Cerevisiae. In this procedure, you have amplification and delta sequences. I hope I will not bore you. I hope I will not be too technical. But I need to clarify a couple of principles, because the functioning of DNA fragments uses a technique which is polymerase reaction, chain reaction, PCR. I would like to explain briefly what it is. In a typical PCR reaction, you have a number of essential components. That is the amplified DNA, nucleotides, which are the small bricks used to construct the new molecules, and primers, which are small sequences of DNA which pair specifically with the sequences to be amplified. And they serve as triggers for the enzymes, which is in this DNA polymerase, and it is thermostable. The reaction normally follows a cycle whereby you have three subsequent steps. First, denaturation of the double helix of the DNA double string, then cooling down to the pairing temperature of primers, and then polymerization. As you can see, these three steps are repeated for an n number of cycles. In the end, this makes it possible to produce an X number of DNA pairs. With n cycles, you have 2 to n pairs of DNA. I wanted to show to you a video, but unfortunately I'm not able to start it. It doesn't start. I'm trying.
mi dispiace, quindi procedo. Detto che quindi la tecnica per ottenere questo...
Unfortunately, we could not see the video. So, having said that PCR is the method which is used, I will now dwell for a moment on the delta sequences which are amplified. They are short, repeated sequences which are dispersed in the Cerevisiae genome in several pairs, because they are components of the transposons, TY1 and 2 types. They are genetic elements that duplicate the transposons, and they are inserted in other points in the genome, and they, in a way, define the evolution distance and the degree of kinship of elements. This type of sequence is present in an X number of pairs in the genome. For S228C, which is the reference element, so S288C is the reference. There you have approximately 300 units delta sequences which are scattered around in the genome. And yet only a small fraction of these sequences can be used for amplification purposes under these conditions with these delta 12 and 21 primers. Out of 300, only 25 can be amplified and lead to PCR. Out of 25, only 11 were confirmed experimentally, which means a very limited number of fragments compared to the theoretically available fragments. Things become perhaps a bit clearer when making an example about chromosome 16 and this triad, because you see that the distribution of products is preferentially on a number of chromosomes. It is not that there is an equal distribution. What happens in chromosome 16, for instance, is that at 62 kilobases, in a space of 1,000 bases, there are 3 delta elements, delta 4, 5, and 6, which are adjacent. The 2 primers, delta 12 and 21, pair in a divergent way. What does that mean? It means that when we launch the PCR reaction I told you about, we potentially obtain 3 fragments, which are the DNA which separates delta 4 from delta 5 from delta 6, or delta 4 from delta 6, which means that we are looking at the interdelta DNA regions, those regions that vary most when elements move and duplicate within the cerevisia genomes. This entails that one strain can be specifically identified when we compare the intra-delta or interdelta profile of the entire genome. If we compare and contrast it with other strains, then we have a possible univocal identification. In this work, what happens is that 4 laboratory strains are analyzed, the black ones, that are kins, because historically they derive from studies and selections that were made in history. And then we have 2 commercial strains. After PCR products are separated based on their size using agarose gel electrophoresis, you have the following. There are 10 or so profiles which were expected out of the potential theoretical ones, and the number of stripes is reduced, meaning that here the pattern, the profile is absolutely unique and can be easily distinguished from the others. Based on these assumptions, we focused then on our goal, that is, we optimized reaction conditions and we worked to devise everything which was necessary to extract genomic DNA and in order to identify the PCR parameters. Then we applied this knowledge and these protocols to a number of strains that were of interest. What we obtained as a result was genotyping of the 32 strains which were of interest for Italiana Biotecnologie. Their comparison led to a number of things. First, 23 identifiable profiles are unique, meaning that they can be distinguished from others. So again, we have 23 identifiable profiles. If we group them by number of strains, 18 strains, the black ones, are absolutely unique, perfectly distinguishable from the other 31. There can be no ambiguity. On the other hand, 8 strains actually had binary identities. You see the light blue one and the other light blue one. They basically overlap, and this overlapping can be due to an identity of strain or to a double isolate. We did not investigate that, but this is what we obtained. And then we had 6 strains which had an absolutely identical profile. Out of 6, one was subjected to the DNA microsatellite analysis, and this analysis actually proved that 5 out of 6 are identical. So the 6 were actually subjected to DNA microsatellite analysis. 5 out of 6 were really identical. After obtaining the molecular identity through genotyping, we focused on production. And we talk about yeast production. As for the molecular identity of the raw material, what happens is that when IB has to go for the production of a certain strain of yeast, it prepares 2 twin dishes. One is sent to Padova, to our lab, and one is sent to the producer. The difference is that in Padova, you can immediately go on with the work while the producer is in a standby situation. We extract the genomic DNA and compare these using PCR with our control reference element. And we prepare a working culture identity record, which is a document where we put down what we found for that strain, and basically we indicate the profile of our sample, stating that there we have that strain and only that strain. This document is then sent to IB, which decides or not to start production. Then, at a second level, once the producer has prepared his work, what happens is that Italiana Biotecnologie receives a sample of the batches produced, and a microbiological or extensive or performance control is performed. And they also prepare 1 dish plate for us, which contains approximately 20 colonies. Out of the 20 colonies that we receive, we take 10 and analyze, say, 50% of the sample of the batch, which is a fairly extensive analysis. We compare and contrast what we obtain from our reaction with the reference element that had been sent in the very beginning. We prepare an identity report where we state whether the sample is compliant with our reference. There are 3 examples. First example, everything is fine and well. There is 100% correspondence and compliance between the two. So the information we give back to IB is a green light. You can accept the batch. And yet, we also had other cases where, for instance, there is a batch which is rich in contaminants, meaning that 50% is having a profile which is a different one. So 5 colonies out of 10. Is it a different yeast? We are not interested in knowing who that is, but the problem is that there is a major presence of another yeast and not the reference one. This is recorded, and IB then will make the decision that it has to make. We have also extreme cases where what we received was totally different from what we had sent. And also in this case, evidently, molecular control entails a problem in terms of acceptance and acceptance of the quality of the batch. That was our contribution, and we think that we have performed good work. Of course, we are working in order to improve it. In our cooperation, we will shortly focus on additional information about a small fraction of yeasts that can be of special interest for Italiana Biotecnologie, in order to have a detailed picture of the sequence. That entailed launching a sequencing program for the genome of a number of strains. And the goal is that of being able of using and exploiting all quality control phases, because the sequencing approach entails, for instance, that you can spot sequences of genes or DNA which are unique for the strain, and this happens. Or you could have a specific combinations of SNPs, that is information which is useful in order to optimize the quality control and our action in the production chain.
Another possible path that we are in the process of exploring is to elaborate new experimental strategies to test some relevant parameters simultaneously. For instance, on top of knowing that from a molecular standpoint, a given strain is what we want to have, we like also to ascertain whether the same kind of reaction can provide us information as to the purity, whether there are other yeasts in the sample considered, or if there can be a correlation with any of the live yeasts, and therefore viability checks could be performed, and that through one single qPCR reaction. But that is something that we're still working on. That concludes my presentation. So I'll leave the floor once again to Paolo, while thanking you for your attention.